Good morning. In this video, we are going to explain document 3 of chapter 7 in the Lebanese program for LS students, the induction of the specific immune response. As explained before in the previous videos, the specific immune response is only found in vertebrates. The immune system of the higher vertebrates allows them to neutralize or eliminate any molecule or any cell representing a danger to the organism. The T and B lymphocytes are the effectors of the specific immune response. They play an important role in maintaining the integrity of an organism. Why? Since they express specific receptors called antigen receptors, which allow them to recognize all the molecules present in an individual. While the B cell antigen receptors or antibodies recognize only the antigen receptors expressed on the surface of T lymphocytes or TCR recognize the antigen associated with an MHC major histocompatibility complex molecule of class 1 expressed on all nucleated cells or class 2 expressed on the surface of macrophages. The specific immune response is triggered when the antigen is taken up by a cell called antigen presenting cell, APC, and presented to the T lymphocytes, mainly to the helper T lymphocytes. So when the macrophage, after phagocyting the microbe, when it presents the microbial peptides in its MHC class 2 molecule to a specific T helper, now the specific immune response is triggered. However, while the non-specific immune response, characterized by inflammation and phagocytosis, develops rapidly at the infection site, the specific immune response must first be induced in the secondary lymphoid organs and not at the infection site. Let's start by some characteristics of the lymphocytes. The lymphocytes are called immunocompetent cells, which means they are mature cells capable of binding specifically to an antigen and triggering a specific immune reaction. The lymphocytes circulate in the blood and in the lymph and stay in the secondary lymphoid organs. After maturation of the lymphocytes in the primary lymphoid organs, bone marrow and thymus, they leave these primary lymphoid organs and they enter the secondary lymphoid organs where they will be activated. The secondary lymphoid organs are mainly the spleen and the lymph nodes. The lymphocytes differ by their function and not by their morphology. You see, the T lymphocytes and the B lymphocytes have the same morphology but have different roles. Here we have another subpopulation of lymphocytes called natural killers. These cells are also involved or important against cancer cells and viral infected cells. They act like the cytotoxic T lymphocytes. Mauger experiment. Mauger has demonstrated in 1967 the induction of the specific immune response and the important role of macrophages in this induction. Its experiment involved the immunization of a mouse with sheep red blood cells with a non-self antigen, the sheep red blood cells. Immunization of the mouse, يعني injection of an intruder, a non-self antigen into an animal, in order to trigger an immune response. So, Mosier has immunized a mouse with sheep red blood cells. So, after immunizing the mouse, we remove its spleen, and we extract from the spleen the macrophages and the lymphocytes. Then we separate these cells into 3 culture media. The first medium contains only macrophages. The third medium contains only lymphocytes, while the second medium contains both macrophages and lymphocytes. Now, the 3 culture media are cultured in the presence of SRBC again. It's the second contact with the antigen for 4 days. And these are the results of the experiment. The question is, interpret the results of document A. Verb interpret, analyze, then give a significance for the result. This indicates what? We can say that we can start by the results. There is no antibodies production neither in the first culture medium containing macrophages only, nor in the third culture medium containing lymphocytes only and SRBC antigens. However, there is production of anti-SRBC antibodies in the second medium when macrophages and lymphocytes are cultured in the presence of the sheep red blood cells. This indicates what? This indicates that the simultaneous presence of macrophages and lymphocytes is necessary for the production of antibodies. So what can we conclude? We conclude that there is a cooperation between macrophages and lymphocytes. So due to the cooperation of these 2 types of leukocytes, due to this cooperation, specific antibodies are produced against the antigen. So we need the previous contact with the antigen plus the presence of both macrophages and lymphocytes in order to induce a specific immune response against a given antigen. So it's the macrophage presenting the bacterial peptide into MHC class II molecule, and this is the T lymphocyte, T helper lymphocyte, having the TCR receptor on its surface. There is a certain cooperation between the macrophage and the T helper. In other words, there is a cooperation and interaction between the non-specific immune response, which is the phagocytosis, and the specific immune response. This cooperation will be detailed in the next video. Thanks for listening.